everyone. Welcome to Gebze Technical University Department of Bioengineering webinar series. Our guest for today's webinar is Assistant Professor Dr. Şebnem EŞSİZ from Kadir University. And the webinar title is Homology Modeling and Molecular Dynamics Study of Stipform Proteins. Prior to joining the faculty at Kadir University, Şebnem EŞSİZ had postdoctoral scholar positions at Lawrence Livermore National Laboratory and Professor Andrew Sally's laboratory at the University of California, San Francisco. She received her PhD in computational biophysics from University of Pittsburgh, following her bachelor degree in chemistry from Koç University, Turkey. Her main research interests are coarse-grained simulation techniques for the study of long-time scale conformational dynamics of proteins, ion channel proteins, ligand-induced conformational chains, and enhanced sampling algorithms for homology-based structure prediction tools. Dear Professor, stage is yours.
Thank you very much. So my title, hello all. My title is Homology Modeling and Molecular Dynamics Study of Stubborn Proteins. I try to pick up a general title, but by the way, my voice is okay, right? I guess so. Stubborn proteins. So what do I mean by stubborn proteins? You're going to see one of them. I'm going to talk about two proteins today, and one of them has a really deep binding site, which is really hard to access, and the other one is an ion channel, which is really difficult to open to permeation, at least computationally. That is why the title has stubborn in it. Now, during all my studies, I always be around the nerve, so that is why I'm gonna describe you a little about how the signal propagation is taking place in the nerve. So there is a resting potential in a normal neuron. That means the charge difference between the inside and outside of the membrane is different. And in a resting neuron, that membrane potential is minus 60 to 75 millivolts. So we can think it like membrane potential is like a battery providing power to drive current when the cell is activated. And when a nerve impulse is transmitted along a neuron, a wave of electrical activity passes along it. Here, just note that it is not. So what I was saying is, the electrical activity is not the flow of electrons. It is just an electrical current. Think it like that. Mainly, there is a transient change in the electrical charge on the membrane surface, and this is transmitted through the nerve axon, and it's called action potential, and it is mainly regulated by sodium and potassium channels. The issue is when you come to the synapse, and synapse is it is an electrical activity. There is a change in the membrane potential along the axon, but when you come to the synapse, it's a physical junction, right? There is actually a small gap of 20 nanometer between the cells, so there is no membrane continuity. Think it like you are in electricity and you come to water. There's a river in between the two nerves. So how are you going to pass to the other side? That is our proteins will take place in that synapse. And this is the signal propagation. So these vesicles, they open, and after they receive the electric signal, they open. Then that electrical signal translates to the chemical signal. Mainly chemical molecules are distributed to the synapse. Then there are these blue small receptors on the other side of the river. They receive those neurotransmitter molecules, and depending on what they permeate, whether positively or negatively charged ions, that cell signal, that electric signal, will be transmitted to the other cells mainly. And the first molecule that I'm going to talk about, the first protein, is acetylcholinesterase. And organophosphates are inhibiting these acetylcholinesterase molecules, such as like chemical weapons, pesticides, oil additives. And let me show you what we are talking about. This is the synapse again, just another picture of that. We have acetylcholinesterase attached to the membrane, and it's breaks down acetylcholine into acetate and choline. So there is uptake back, taking back to the pre-synapse. And if you inhibit this acetylcholinesterase by chemical regions, then you're gonna have, based on the exposure levels, you will have cognitive deficiencies, seizures, paralysis, and death. What is happening when you block this protein? Then you cannot break down acetylcholine. Then the concentration of acetylcholine is increasing in the synapse. Then the receptors are overly activated. That is the you have those seizures, stuff like that. Okay, this is an antidote kit. This is my work from that national lab, actually. It is a biodefense lab. So we have these for years. We have these antidotes. So when somebody is exposed to a nerve agent, then you just inject these oximes, and 2-PAM is one of them, and some countries carry atropine to reactivate the acetylcholinesterase. However, the issue is 2-PAM is not significantly affect all the nerve agents that we know, like SOMAN, and no single compound effective for all chemical weapons. And finally, current reactivators do not effectively penetrate the blood-brain barrier, actually. So this is the protein that we are talking about. It is present in all nerve synapses and neuromuscular junctions. And wild-type enzyme is a tetrameric form. And I showed you in the picture, it is linked to the membrane. It is highly conserved across mammals. And in our calculations, we're going to use monomers, actually. Let me show you the binding site. The peripheral anionic site is like the gatekeeper. It is beginning of that gorge and may function as gatekeeper. Then there is the catalytic triad. There is the choline binding site, which is around tryptophan 86. And oxyanionic holds the carbonyl oxygen in place. So this binding site is for acetylcholine. And here, if you can see it, it's a basic addition-elimination reaction. This is the serine residue in that binding site. This is acetylcholine, and this is the acetyl enzyme. Acetylcholine is done in a next step. It will break from here also. So the enzyme will be reactivated mainly. So this is the idea. We have the serine residue, and acetylcholine gets adducted to the serine residue. Then acyl enzyme is formed, and then a water molecule is attacking from the carbon center. Then the enzyme is reactivated, so it keeps working. What is happening in the organophosphate case? When the serine residue again is adducted, just like acetylcholine. However, after this pinocoyl R group leaves, the enzyme gets a resonance structure, and once it is aged like that, it doesn't get reactivated. Okay, that is the inhibition mechanism. And we did some first, we tried to calculate the energy barriers for SOMON, serine, these are the nerve agents, and the regular neurotransmitter is acetylcholine. So this is a QMMM calculation. Mainly, we have 34 residues in the quantum mechanics region, plus water molecules. And we use the in-house program in the Livermore lab. It's called MGMOL, based on density functional theory. And we have 823 wave functions. And it takes 8 hours on 640 CPUs on a Linux cluster to calculate transition state of this molecule. And when we look to the energy barriers, these are quantum calculation values. There are 2 transition states for the SOMAN, and the adduction, as you can see, is much lower than the acetylcholine. So it gets much easily attached to the serine residue when compared with the acetylcholine. Now, oximes, just like that 2-PAM that I showed you, can reactivate this enzyme. So what our aim is, we are trying to find better oximes than the 2-PAM. But the issue is we have all these bulkier oximes. However, when we dock oximes to that binding site, since it is really deep in as the gorge from the surface, it cannot get that close to the serine active center actually. So there is a 7 angstrom difference here. That means that nucleophilic attack cannot happen. And here is a molecular dynamics movie actually. It's a simulation that it shows here is the oxime and the yellow is the serine residue. So the molecule is trying to get into that binding pocket. And you are seeing all the rotation stuff like that. Here is the oxime. And it cannot come close to that binding site actually. So in the next step, what we did was we ran 40 different MD simulations, molecular dynamics simulations, of the APO and SOMON-adducted acetylcholinesterase to see is there a population of adducted protein that is sensitive to oximes more than the APO form. And I'm going to explain more like what we are looking for. We are looking for different binding pockets or entrances to the molecule. Molecular dynamics is a simulation technique which uses Newton's second law of motion mainly. Force is equals to mass times acceleration. And from here, if we know the initial positions of the atom, and we know those initial positions from the X-ray structures, then if we know the temperature, and if we have a force field, which all these simulation packages comes with these force fields, which are dependent on bond, angle, dihedral, and non-bonded terms, then we can calculate that acceleration at each time step, and we can see the dynamics of the molecule, actually. So when we look from the side of the molecule, this is the gorge entrance. That is what I mean by the stubbornness. It is really deep in the, it's like a sphere, and there is an entrance to that molecule. I start to hear one of your mic. Okay, so after those 40 times 50 replicas of those simulations, 50 nanosecond simulations, we look to the differences in between the APO and SOMON-adducted simulations. So the first thing that we look to that gorge mouth, what is happening there, and we are seeing in the SOMON-adducted case, when we compare with the APO form, the gorge gets much larger than the APO form. There is also a side entrance. So in addition to that main gorge entrance, there is a different place that the molecule can get through. And that is also, in the SOMON-adducted case, it is getting much larger than the APO form. So there might be an alternative entrance in addition to that main entrance to the molecule. We also check all the tunnels from that serine residue to any opening at the protein surface, just like here you are seeing one of them. And the numbers, the results show that the influence of SOMON-adduct on the tunnels in the distributions is getting more to the left-hand side for the SOMON case. This is the principal component. Should I explain it? Like when we do the molecular dynamics simulations, we are going at each time step, we are going 1 femtosecond fast. So there is so much small vibrations in the molecule, but what we are looking is more large scale motions. So here what you are seeing is called principal component analysis, and that is a technique to remove that noise from the system so we can see the larger scale motions. Here I'm not showing those principal components, but in the second part I'm gonna show that also. These graphs, these ones are showing the correlations which are calculated from those principal component analysis. Although the gorge entrance area is getting larger, the correlations in between different regions. When I say correlations, when one part is moving, if the other part is moving correlated to that, they are sort of doing the same, the motion in the same scale. If they are anti-correlated, they are doing it on the opposite direction. And if there is no correlation, that means they lost the connectivity in between them. So what we are seeing in the somnoduct case is mainly the correlations are getting reduced. So here we tried that side entrance. Since we found that new entrance to the molecule, we tried to dock the oxime from side entrance instead of the gorge entrance area. Now we have 4 angstrom distance in between that soman and the oxime. So this might be the alternative binding route for the soman molecule. So this is the conclusion for the first part. Soman adduction is more favorable than serine or acetylcholine. Soman adduct increases a different door, which is called the side door area. So that might be the alternative pathway into the active site. And when soman is bound, then it decreases the correlation motions from the distant side of the protein. So it is sort of killing that talk in between different parts of the protein mainly. We are now working on the different nerve agents actually, serine and tubulin. These molecules are now, we are comparing that to soman. Now in the second part, we are again in that river, and right now, let's say we reach to neurotransmitter molecules reach to the other side, and here are those receptors, right? When if they can reach to the other side of the river, then they're gonna go through those receptors. Here, I'm not gonna talk about specifically the acetylcholine receptor, but now we're gonna change the receptor, and it is activated by aspartate. And these are ion channels, receptor complexes. And it's called ionotropic, since they permeate ions, glutamate receptors. It's a family of proteins, actually. It has a vital importance in conscious and maintaining normal brain function. And again, overstimulation of these receptors end up with stroke, epilepsy, Alzheimer, and Parkinson's disease. Actually, memantine is the approved drug for Alzheimer's, and it is acting on the NMDA receptors. Now, as I said, there is the ion channel, which is normally closed in the resting state. And there is the ligand binding domain. Then there is an amino terminal domain at the very end of the protein. Here, the natural ligand will bind to the ligand binding domain. And when the ligand binds, the ion channel part will open. Then ions will go from outside to the inside of the channel. And we can have positive amino terminal domain is an interesting domain in this family of proteins. In the other members of the glutamate receptors, it doesn't do any function, but in the NMDA receptor, it regulates the activity of the ion channel, either positive or negative. So when some molecule binds to the amino terminal domain, it affects the permeation of the channel also. And structure of NMD is a heterotetramer, consists of 2 dimers of diamonds with a 2-fold axis actually. So there is GluN1 and GluN2 subunits. And GluN2 subunits is the glutamate binding domain. So there are 2 glutamate molecules which binds to that ligand binding domain. GluN1 subunits is binding glycine. And that is also required actually for the activation of the channel. So there are 2 glutamate molecules binding to the N2 and 2 glycine molecules binding to the N1 subunits. Now, did you hear about the Nobel Prize in medicine recently. Just 2-3 days ago. They are for solving the structure of ion channel proteins again, but the proteins is not the ligand gated ion channels, but one of them is mechanosensitive ion channel, for instance. It answers, like it reacts to the curvature in the membrane, then it opens to permeate ions. And the other one is heat sensitive. It is again an ion channel. So why are these people, like when they solve these X-ray structures, actually, when they understand the structure of the protein, they are getting Nobel Prizes. It happened in 2003 also. It was a potassium channel then, but right now I lost my... Do you see? I hope there's no problem with the slides. So the problem is resolving these structures are really difficult. Like, since they are embedded in the membrane and they're also very flexible in the outside, in the globular region. So it becomes really hard to crystallize these proteins because you are taking them from their natural environment, actually. And so the resolutions and the structures are always problematic. So until 2014, there is only one intact AMPA glutamate receptor, which is a different member of the family from the same family. But then in 2014, there were two resolved X-ray structures. One of them was from frog, the other one is from rat. And the resolutions are not bad. However, the structure, right now we are looking to the ion channel part. So this is the channel which permeates ions, and we are seeing the most restricted regions. And this is the red structure, actually. Do you see the bottom of the channel is really not resolved well? That is the reason why you are not seeing the channel clearly, because there is some not inconsistencies, but uncertainties about those atomic resolution parts at the end of the channel. So by using these two structures, our aim was to model the human structure. And let me show you the issues. Like, this is, at this step, we use homology modeling. And in the homology modeling, you try to find a proper template for your unknown structure, and you developed a model based on the structure of that template. On the x-axis, we are seeing the sequence identity, and on the y-axis, there is the structural overlap. So even the sequence identity in between your template and your target sequence is low. For instance, even if the sequence identity is around 20%, the structural similarity is much higher than that. This is the idea of the homology modeling. So although the sequence is not that well conserved, structure is much better conserved than sequence. That is the main assumption. Actually, we are seeing here that assumption is also correct. And here we took those red and not red, the frog structure, and analyzed that more. And we are seeing there are large loop structures that are not resolved on those X-ray structures. Also, several loops in the human structure are not found in those animal structures. That is the other issue. And here I am just showing the dimeric structure. It's actually a tetramer. So these loop structures that I highlighted with red and blue are not present in the X-ray structure. So we need to develop a modeling scheme for efficient modeling. What we did first, we took the template structure and we modeled the human structure with the loops based on that tetramer structure. Then we check our model's quality with ProCheck and Ramachandran plots. Ramachandran plot showed us that only 77% of the model is in the allowed regions actually. While when we look to the template structure, the allowed region percentage is 94%. So that means actually, why are we looking to the template structures Ramachandran plot is, we can get as good as our template, right? So we cannot get any more information from what we have in the template, but we are far below that also. Ramachandran plot for the loops are making it much lower. So what we did was, we took this, we are doing these things in modeler, and we chopped those loop regions and modeled this structure as a tetramer without the loops. And then we saw that project, the Ramachandran results are getting much better. So we came to a 90% region. And then we built those loop structures on with different algorithms. One of them is Rosetta, and it is more like ab initio type of method, which means that you are building those loops from scratch. And in modeler, also we did some loop modeling. And in modeler, it is more knowledge, it's a hybrid in between ab initio and knowledge based. And we try to compare those loop structures that we built. So for each region, for each missing loop, we built different models and evaluate them. Here, this is the modeler scoring function, and x-axis is the amino acid index. So here, the models are same other than the loop regions, for instance. We are just looking to one subunit. And red one is where we started. The lower the score, the better the model gets. So we compared the loops that we modeled from Rosetta and modeler, actually with modeler scoring function. And we saw that Rosetta is giving better results in terms of the scoring function. So we used, for each model, we built 100 different loop structures, and we picked the best ones according to the modeling scoring function. Then for the simulation part, we have an unequilibrated POP-C membrane. Then we insert our structure into that membrane. And final system, after adding the ions and everything, it is 230,000 atoms. It is actually more than that because we need to make the box larger, and it is around 300,000 atoms now. What we do is, in an MD, this is the molecular dynamics simulation again. This is the simulation box. And we have the protein molecule, lipid molecule. There's actually water molecules surrounding this box, but we didn't show them. This is just the ions that you are seeing. And here first we are trying to imitate the real system, right, in a simulation box. So what we do is we first equilibrate that POP-C level. Oops, sorry. Then we release the whole molecule. First we restrain the backbone, then we release the whole molecule. And of course, everything is not that smooth, so we came across problems. Here you are seeing the two glutamate molecules on the ligand binding side. And let's see the initial step. We remove the lipid and the water molecules here. Now, if you watch, this is after like 10 nanoseconds, I think, one of the molecules will leave the binding pockets and it goes outside the box, then it comes back and it finds its correct place again, but we put some more restraints to these glutamate molecules in the beginning to make that equilibration smoother also. So at the end, we run this 1000 for the APO form, and for the glycine and glutamate bound structures, we run 1000 nanosecond simulations and we try to analyze the differences. Actually, what we were hoping was, in the hollow simulation, we will see whether we will see an opening in the ion channel part. Again, as I said, this is the transmembrane part that you are seeing, but the extracellular domain is really large. and you think about like two small molecules is attaching here, not attaching, interacting with that, and then that allosteric effect will be distributed to very far side of the protein. So in those simulations, we saw a slight opening. This is the red one, is that most like the transmembrane segments are coming together on top of each other at the top of the channel. And if it opens, it will open from here. And we are seeing an enlargement, a widening in that area, but it is not that significant. By the way, what is our constraint? Like, there will be ions passing from this, right? So in order to call it open, it should let sodium ion or calcium ion should be able to pass from that region. Think about that scale. So this is called a pre-open state, actually. So we couldn't go to the fully open state at the end. And there is a bottom girdle. There is another segment, another restricted region. That region didn't even feel that effect. It is worse than the top one, but there is a little this difference in the distribution again for the hollow simulation. However, we saw different dynamics in the upper part of the protein, like when this is the amino terminal domain, and the red one is for the apo, and the green one is for the hollow simulation. So this is getting more compact in the hollow simulation. By the ligand binding domain, each subunit, the yellow one, there is a closure motion on top of ligand, but then it gets more compact also in terms of the ligand binding domain. And this is the interesting part, that the distance between the centers of amino terminal domain and ligand binding domain when it gets smaller, those closure motion pulls the linker region. So this is linked to the transmembrane segment. And then for the hollow simulation, that linker gets longer. That means there's a pulling motion. So we are starting to see, so this loops will pull those helices, and they are getting longer in the hollow simulation. And here is what I was trying to talk about, principal component analysis. So from all those fluctuations that we saw here, we are trying to capture the large scale motion by removing this noise in the system. And we end up with motions like this. The right-hand side is the hollow simulation. The left-hand side is the apo simulation. Now, when we come to the GluN2A units, these are the glutamate binding subunits. You will for sure notice the scale of the motions is much larger in the hollow simulation. And yeah, the right-hand side is the hollow simulation. Now, we also check the tilt angle in between the apo and hollow simulation. So here you are seeing the apo simulation with respect to the initial structure. And here when the glutamate is bound, so there is like, especially for one of the subunits, there is that tilt in the extracellular domain. In the cryo-EM images of these proteins, there are different clusters, and one of the clusters is showing the same type of that tilt angle. So the protein is doing this motion, and it is visible from the cryo-EM images also. And it is doing it for the in the glutamate subunits, actually, more than the glycine one. So this is what I said. Glutamate binding subunit is displaying larger correlations and rigid body motion. Amino terminal domain displays a tilt motion more in the glutamate binding subdomains again. Overall, GluN2A subunits display the more significant conformational change with respect to GluN1, and especially the linker region, M3S2, is almost 12 angstrom longer for GluN2 subunits in the homology simulation, while there is no significant difference in GluN1. Now, in the next step, this was published, then we try to understand the significance of that amino terminal domain mainly. And what we are planning to do, what we are doing right now is, this is the ion channel part. It is aligned with amino acids that we know, sorry guys. And we are doing some mutations exactly where that most restricted region is. This is done experimentally also, the mutations. So we know that it opens the channel experimentally. What we are trying to do is in the structure, in the computer, trying to open those structures. Then we're gonna have and here you are seeing those openings. The cyan ones are the mutated ones. And after we take back, our plan is taking back these mutations and still capturing the open structure. Then we will dock small molecules. In addition to the ligand binding domain, we are planning to dock molecules to the amino terminal domain and see their effects in the transmembrane segment. So that way we might be able to understand the role of the amino terminal domain specific to NMDA subunits. And here you are seeing that mutation effect actually. This is not the directly mutated site, but just top of that. So this is the apo structure, and this is the region where we have the mutation. Then the pore is opening from there. So this is, as I said, two stubborn protein story. And for the acetylcholinesterase, we worked with Melis Gencel. And for the NMDA one, I worked with Mohammed Ayhan, and Burak Servili is my PhD, also Melis. And we are doing all these calculations in the Livermore National Lab, actually. We are using their supercomputers, and these are our collaborators from there. And just to give you an idea, I also worked on the homology modeling methods. So I didn't talk about methods, I just talked about proteins actually today. But I also worked with, when I was talking about these alignment issues, the gap between structure and sequence alignments, if we can incorporate that structural information to the alignment problem, then we will reduce the problems in the alignment. That is an algorithm that we are trying to develop. So on the fly, we are trying to get better alignments by doing genetic algorithms moves to the alignments, and then building models, evaluating them, and going back and doing these. So it is like sampling in the sequence space instead of sampling in the protein structure. This is one homology modeling project. Then I also have these rigid body dynamics method development. Mainly in that project, we decompose proteins into rigid segments. Again, this is just coming from that correlation analysis also. The parts that moves together will be decomposed into rigid bodies. And if the atoms are moving more independently, then they're gonna live in all atom detail. And then the motions of the protein will be simulated in this reduced subspace. So some of the protein parts, for instance, we know that benzene rings are moving together, right? The atoms in the benzene ring. So they will be taken as rigid bodies. That will save us time from the computation of the forces at each time step. And the aim is making it faster. That is done, but adding a heat piston to the rigid body dynamics is still an ongoing research. I also have some studies with experimentalists about docking studies directly for different proteins. That's it then. Thank you for listening. I can take any questions if you have.
Dear Professor, thank you very much for your impressive presentation.
Thank you, guys.
If you have any question, please click right now.
I'm seeing some of my students also. Merve is, she's not even in Turkey, I guess. Okay, any questions? I got distracted.
Is there any question? I think there is no question.
Okay, guys.
Okay, thank you again, dear professor. If there is not any question, we could end this webinar right now. Thank you again.
Bye bye, guys.
See you.